it feels like we're just drowning in information. I mean, if you try to keep up with every single scientific paper published, you'd never sleep. But every once in a while, research emerges that, well, it doesn't just refine an old concept, it reveals a fundamental piece of biological machinery that we simply did not know existed. And that moment happened recently with a truly groundbreaking paper by Wang and colleagues. They identified an entirely new way a cell can die, a form of regulated episodic death. It's a finding that could genuinely change the map for how we understand everything from infection to cancer.
Absolutely one of those papers.
Yeah.
And, when you identified a whole new mechanism of death, the field needs new terminology. So they define the overall process where the cell literally bursts open as mitoxyperilysis.
Mitoxyperilysis. Okay.
And this specific, very localized damage from the mitochondria that delivers that final blow. They've named that mitoxyperiosis.
Mitoxyperilysis and mitoxyperiosis. Got it. Okay, so let's unpack this. Our mission today is pretty straightforward to explain the very precise, very unique synergy that's required to trigger this, why it's so different from all the pathways we already know, like apoptosis or necroptosis. And what I find most fascinating, how a really surprising molecular gatekeeper, this protein complex mTORC2, actually orchestrates the whole thing.
Right, and the single most critical takeaway, I think, is that synergy you mentioned. This whole pathway only kicks off when you combine two specific types of stress. They call it innate immune activation and metabolic disruption, or for short, IIAMD.
IIAMD.
And you really should think of this combination as a perfect storm. It's a storm that happens constantly in some of the most critical diseases we face, sepsis, chronic inflammation, and especially in the really hostile environment of a growing tumor.
So why both signals? That's what I'm stuck on. I mean, normally if a cell is under attack, an immune signal should be enough to trigger something like, say, pyroptosis, right?
Precisely. That was the first question. They started with the classic immune cells bone marrow-derived macrophages, or BMDMs, to see if one signal would be enough. So first, they just tried metabolic disruption. They basically starved the cells of carbon. They survived. Then they tried just the immune activation ligands, things like LPS, which mimics a bacterial infection, or R848 for a viral attack.
And the cells were fine then, too.
They survived the initial challenge, yeah. They didn't burst.
But the moment they combined them, say LPS plus the carbon starvation?
The results were dramatic. Only that combination, the IIAMD, induced this really robust serolytic cell death. You could actually measure the membrane rupture by seeing all the cellular contents, like LDH and HMGB1, just spill out into the medium.
So that confirms it. The metabolic pressure isn't just a side effect, it's a non-negotiable part of the trigger.
Absolutely. And they ran the critical check. If they took those dying cells and just gave them back some basic fuel glucose, glutamine, even pyruvate, the cell deaths just stopped. It was completely blocked. So if you fix the energy crisis, the cell basically refuses to die by this mechanism, even when the immune alarm is blaring.
That is a beautiful demonstration in vitro. But, this is a deep dive, not a lab meeting. Let's talk about the real-world relevance. Did this synergy actually hold up in a living organism?
It did. And the finding has huge clinical implications. They used a mouse model for septic shock, which is just this massive immune challenge. They found that if they pre-fasted the mice, inducing that metabolic disruption before hitting them with the LPS challenge.
Oh, I think I see where this is going.
The animals had much higher mortality, and they died faster than the mice that weren't fasted.
So the pre-fasting literally created the vulnerability.
Exactly. That group showed all the markers of massive cell and tissue damage, LDH, ALT, AST just skyrocketing, indicating cells were bursting all over the body. It really confirms that this two-pronged attack is what triggers this lethal process, not just in a dish, but in a whole system.
Okay, quick recap then. The cell needs both the immune alarm to go off and an energy crisis at the same time. Neither one alone is enough to trigger this unique form of destruction.
That's the key.
So now we know how to trigger it. Let's figure out what it is. If the cell is bursting, that points to a lytic death. But we already have pyroptosis, which uses gastromin D, and necroptosis, which uses MLKL. Why isn't this just a variation on one of those?
Yeah, it is the essential next step, right? Defining what makes it new. And they were incredibly thorough. First, they ruled out caspases. This pathway is completely caspase independent.
Okay, so that immediately crosses apoptosis off the list, and the classic caspase 1 form of pyroptosis.
Right. Using the pan-caspase inhibitor ZVA did nothing. The necroptosis inhibitor NEC1s also failed to prevent the death, but then they went further. They ruled out the primary executioners of necrotic death themselves. The pathway is independent of all the known pore-forming proteins.
The gastramins and MLKL.
The whole family. Gastramin D, gastramin E, MLKL. They used cells that were genetically knocked out for these, so cells that should be completely protected from other lytic deaths.
They still died.
They were still perfectly susceptible to mitoxyperilysis.
So no caspase, no known pore formers. What about some of the newer ones like pyroptosis? That's driven by oxidized lipids or oxidative.
Check those too. The pyroptosis inhibitor, ferristatin-1, offered zero protection. And they ruled out oxidative by knocking down its key sensor, KeyAP1. Again, no effect. This isn't just a variant. It's a completely separate cascade.
Okay, so if it's not using any of the known exits, we have to ask, what's the universal ingredient? What's the thing that actually drives the damage?
The common driver they landed on is oxidative stress, and it's specifically linked to that metabolic crash. Their metabolomics data showed that the IIAMD trigger caused this massive metabolic shift, and the most notable change was a huge drop in glutathione, or GSH.
And GSH is basically the cell's master antioxidant, its primary defense against oxidative damage.
It's the master reserve, exactly. When a cell runs low on GSH, its ability to neutralize all those toxic byproducts of stress just collapses. And the proof was simple. They just added GSH back to the system from the outside.
And the cells were protected.
The cell death was significantly reduced, yeah. And this internal weakness, this lack of defense, is what allows the mitochondria to get damaged. This part, at least, uses some familiar machinery. The BH3 domain proteins,
BX and BX,
BAK and BAD, they showed that cells genetically deficient in those proteins were protected from the mitochondrial damage. So these are the critical upstream regulators that punch the initial holes in mitochondria, which in turn leak out all the deadly oxidative stress.
And here's where, for me, it gets really interesting. This is where the paradigm shifts. Okay, so the mitochondria are damaged. They're spewing out reactive oxygen species, ROS. How does the cell die from that? For years, the focus has been on the chemistry, just generalized oxidative stress. But this paper proves it's all about the mechanics. It's about physical location.
It is all about the mechanics. The process they named the mitoxyperiosis shifts the focus from the amount of ROS to the delivery of the ROS. They used live imaging, and what they saw was just stunning. The stressed mitochondria didn't just float around, they actively moved and then stably localized right next to the plasma membrane, and they just stayed there for a really prolonged period, often more than 20 minutes, right before the cell burst.
And that prolonged contact is the physical key to the whole thing.
Precisely. Because ROS are so short-lived, they're incredibly unstable chemically. They can't just diffuse across the cell and cause damage randomly.
It's not a chemical cloud filling the room.
No, it's more like a blowtorch.
Yeah.
By forcing the damaged mitochondrion to maintain that prolonged, intimate contact with the membrane, the cell creates a localized oxidative battlefield. They saw this massive accumulation of lipid damage, things like 4-HNE adducts right at that contact site. And that local degeneration is what causes the final burst, the mitoxyperilysis.
So the cell isn't being generally poisoned, it's being fatally injured by a persistent physical touch from its own failing organelle. It's like the damaged mitochondria are being held hostage at the membrane until they burn a hole through it. What on earth controls that forced, lethal proximity?
So to find that master regulator, they did a small molecule screen. And they found that inhibiting mTOR kinase, a classic regulator of cell growth, was highly protective.
Oh.
And they narrowed it down. It turns out it's the mTORC complex, the one regulated by a protein called Richter, and not mTORC1 that promotes the cell death.
Wait, mTORC2, now that is counterintuitive. Isn't mTORC2 usually associated with survival and maintaining cell structure?
Yeah.
Why would a growth promoter be the one that facilitates the cell's destruction by paralyzing it?
That's the brilliance of the finding. They showed that the mTOR activation and the mitochondrial damage were separate events. mTORC2 wasn't causing the stress. It was acting downstream as an execution helper. And its role is, as you said, astonishingly mechanical. It promotes mitoxyperilysis by suppressing cytoskeletal activity.
So, let me see if I have this right. It's like the cell's internal highway patrol, the cytoskeleton, is supposed to move the burning car, the damaged mitochondrion, away from danger, but mTORC2 just froze all the traffic lights, keeping that burning car parked right next to the fragile membrane.
That is an excellent analogy. The paralysis specifically involves shutting down Rho-OA activity, which is the little protein that organizes the actin filaments needed for movement. The cell literally loses its ability to fight back and physically move the dangerous organelles away.
And when they broke that paralysis with the mTOR inhibitor, what happened then?
When they treated the cells with taurin-1, the mTOR inhibitor ROA activity came back online, the cytoskeleton started moving again. The cells started actively forming these sheet-like projections, lamellipodia, right at the contact site.
So it was fighting back.
It was literally pushing and pulling the damaged mitochondria away from the plasma membrane, preserving its own integrity. And they proved this was the key by then adding an actin disruptor, cytoskeleton D, that immediately blocked the protective effect of the mTOR inhibition. The mechanical defense was absolutely essential.
Okay, let's pause and summarize this. This is a complete novel pathway. First, you need the synergistic trigger. Innate immune activation plus metabolic disruption, IIAMD. That trigger causes an internal collapse, depleting glutathione and activating BX and BA, which damages the mitochondria.
And that damage only becomes lethal because the mTORC2 complex then steps in as a mechanical gatekeeper. It suppresses ROAA and paralyzes the cytoskeleton. This paralysis is what ensures that damaged mitochondria can maintain that prolonged, lethal, physical contact with the membrane, which causes a local oxidative burn and ultimately the cell bursting.
That is just a phenomenal piece of work, and it immediately makes you wonder about applications, especially when you think about the tumor microenvironment.
Exactly. The TME is a perfect breeding ground for IIAMD. Tumors are often starved for nutrients and are highly inflamed. This pathway gives you a way to specifically weaponize the body's own inflammatory response against the tumor, as long as you also hit that metabolic component.
Did they actually test that strategy in a cancer model?
They did. They used a B16 melanoma mouse model. They combined an intratumoral LPS injection, that's your immune activation, with pre-fasting for the metabolic disruption.
And it worked?
The combined treatment significantly reduced tumor size and induced massive necrosis, far more than either treatment alone. They successfully turned mitoxyperilysis against the tumor. And, crucially, they confirmed the mechanism was the same. They used tumor cells that were deficient in Richter, the mTORC2 regulator, or ECTR, the mTORC1 regulator. The tumors lacking Richter were partially resistant to the treatment.
Proving that the cytoskeletal paralysis controlled by mTORC2 is the key vulnerability.
That's right. It connects the whole story, from the petri dish to the tumor model.
You know, this sounds like a sophisticated mechanistic update to some old ideas. I'm thinking of things like Coley's toxin from the 19th century, which used bacterial products to induce this huge immune response against tumors. Maybe now we know why that might have worked sometimes. It was the missing piece. You also needed the metabolic stress of the tumor environment to create the IIAMD synergy and make the immune response lethal to the cancer cell itself.
It gives us a new level of precision. Instead of just trying to use general ROS scavengers, which have historically failed in the clinic, this work highlights that the problem isn't just the poison itself, it's the subcellular localization of the poison source.
We're moving from chemistry to cellular engineering. We're not trying to clean up the entire chemical spill. We're trying to fix the traffic controls so the cell can physically remove the danger before it breaches the perimeter.
Right. Targeting mTORC2 or its downstream machinery offers this really unique therapeutic avenue for cancer and maybe other chronic inflammatory diseases. It's a profound shift in thinking.
So we have to leave you with this final thought, building on this incredible mechanical insight. If a cell's own cytoskeleton is what actively protects it from being fatally damaged by its own stressed organelles, how many other seemingly distinct human diseases, from neurodegeneration to chronic inflammation, might actually be linked not by a shared chemical problem, but by the simple mechanical failure of subcellular defense?
A provocative question indeed. It really makes you realize that sometimes where the molecules are matters a whole lot more than how many of them there are.
Thanks for diving deep with us. We'll see you next time.